Hi, I'm Miss Joyce and I'm a chemistry teacher at the Sacred Heart of Mary Girls School. I'm also a teacher of the Applied Science course, or to call it by its full name, the Level 3 Extended Certificate in Applied Science. The Extended Certificate is an advanced Level 3 qualification which is equivalent to one A-level. So you can take it alongside two other subjects and perhaps EPQ. Applied Science is taught by three subject specialist teachers in both Year 13 and Year 12 and is made up of 6 modules. 3 of those modules are coursework based and the other 3 are externally examined. This course also allows you to resit the exams as you have 2 exams in June of Year 12. These are the 6 modules. Unit 1, 2 and 3 are taken in Year 12 and Unit 4, 5 and 6A are taken in Year 13. I've asked 2 of our current Year 13 Applied Science students to give you a rundown of each of the 6 modules. Unit 1, Key Concepts in Science.
Unit 1 is an externally examined module where you study topics in biology, chemistry and physics. The content builds on your GCSE knowledge, but you do not need to know the science in as much depth as you would at A-level. Top tip, keep your GCSE knowledge up to scratch over the summer, otherwise it will be really hard work.
Unit 2, Applied Experimental Techniques.
Unit 2 is the coursework module which you complete in Year 12 and it focuses on 6 short experiments based on a Unit 1 syllabus, 2 for each subject, which offers you the opportunity to develop practical skills in biology, chemistry and physics, getting you ready for practical work in university and in the workplace.
Unit 3, Science in the Modern World.
Unit 3 is called Science in the Modern World. It is an external examined unit with a pre-released case study. It links to all the other modules and helps to develop your analytical, evaluative and critical thinking skills, essential skills for scientists. It also gives you a chance to look at possible careers in science available to you, for example, a radiographer, zoologist or toxicologist.
Unit 4, The Human Body.
Unit 4 is based on the study of the human body. You'll develop an understanding on the human anatomy and physiology, specifically the digestive system and diet, the musculoskeletal system, oxygen transport within the body, and the structure and function of the nervous system. Unit 4 is assessed with a written exam lasting 1 hour and 30 minutes.
Unit 5, Investigating Science.
Unit 5 is another coursework module. We investigate the electrochemical cells and vary their conditions. That's including temperature, the concentration of the electrolyte, and the type of electrode. The investigation links to manufacturers of smartphones and any other electrical devices, as electrochemical cells are the basis of batteries. There are lots of independent practical work, which again will help you prepare for university.
Unit 6, Microbiology.
The aim of Unit 6 is to develop knowledge and understanding of key microbiological concepts and techniques used when working in biotechnological industries. Microorganisms are vitally important in the modern world we live in. We investigate their structures, functions and adaptations. In natural habitats, microorganisms play a crucial role in the decomposition of organic matter and the recycling of nutrients. We practice techniques to grow, identify and isolate them. Microorganisms also cause disease and are responsible for millions of deaths every year, despite ongoing advances in medicines and healthcare.
We're going to have a go at the second half of the Chemistry Unit 2 practical together. We have been given contaminated river water. Here it is. And it's believed it contains copper 2+ ions, which give it its blue colour. It's our job to use a technique called colorimetry to find out what the concentration of the copper is. Why do you think it's important that we find out the concentration of the copper ions in the river water?
Copper 2 sulfate is harmful to the environment at concentrations greater than 1 mole per decimetre cubed. If the contamination of the river continues, it could lead to accumulation in soil, plants and animals. Most animals and plants are not adapted to live in areas of high copper concentrations, and therefore this could cause a decrease in biodiversity. It's also a concern for farming and agriculture, as crops don't grow well on copper-rich soil. Another skill you'll develop in this course is writing risk assessments, and referencing CLIAPS. This is one of their student safety cards, which are available as free PDFs online for you to use. Here I have a cuvette with distilled water, and the other contains the contaminated river water. First, I zero the colorimeter with the distilled water with the first filter, 440 nanometers, and then I test the river water. I do this 3 times to improve the reliability of our results, and then I repeat the process with all the filters and record the results in my results table. Here are our results from our preliminary investigation to determine which filter wavelength we should use. I've calculated the mean, and we want to choose a filter which will give us the most accurate results. From looking at the mean, which filter do you think that will be? It will be the filter with the highest absorbance, as the high value will lower the percentage uncertainty of the data. So, were you correct? We will use the 680 nanometer filter, as it should give us the most accurate data.
To do this, I'm going to measure the absorbance of different concentrations of copper 2+ ions. Then together we'll plot the calibration curve. We'll plot the concentration against the absorbance and draw a line of best fit. Here are my 6 cuvettes with the 6 different concentrations of copper sulfate solution. 0 moles per decimeter cubed, 0.2, 0.4, 0.6, 0.8 and 1 mole per decimeter cubed. Hopefully you can see how they get lighter in colour as we go down. This means we should see a high absorbent value from 1 mole and see it decrease as we move down.
Here is the data collected for the calibration curve. Pause the video to calculate the mean values. Watch out for anomalies. Okay, let's check your mean values. 0.0 moles per decimeter cubed is also 0.00. 0.2 is 0.44. 0.04 is 0.9, not 0.97. As result 3, 1.23 is an anomaly. It doesn't fit the pattern, so we exclude it from our mean. 0.6 moles per decimeter cubed is 0.93. 0.8 is 1.59, and 1 mole per decimeter cubed is 1.99. When you look at the data as a whole, you should also spot another anomaly. Where is it? Hint, it's one of the means. The mean value for 0.6 moles per decimeter cubed is an anomaly. It doesn't increase as much as we would expect it to. What could we have done to improve the accuracy of this data set? We could have discarded the data and repeated the experiment for that concentration. I haven't done that, but doing it would make your data more valid. Your analytical and evaluative skills will really develop through the course, preparing you for work or university. Also notice how all the data is to 2 decimal places. It's scientifically correct to have all the data at the same number of decimal places in a table. Your next job is to plot your calibration curve, with concentration of copper sulfate on the x-axis and mean absorbance on the y. You could do this on graph paper or on Excel or Google Sheets. In Unit 5, we use Google Sheets to produce all our graphs, as part of the module is about using IT to support analysis. Pause the video while you plot your calibration curve. Right, let's check your graph. You'll get 1 mark for labelled axes with units. A second mark for using an appropriate scale. A third mark for points plotted correctly. And a fourth mark for your line of best fit and your anomaly circled. Again, the ability to plot a scientifically correct graph is a key skill you'll develop throughout the course, which will be really useful for scientific careers and study. From your calibration curve, you can then calculate the concentration of copper 2+ ions in the river water. Here's the result for the river water using the 680 nanometer filter. From this, you draw a line from 1.2 ARBs to the line of best fit and down to the x-axis. This enables you to read off the concentration of our unknown sample. Do this on your own graph and find the concentration. Here I've zoomed in on my graph so we can see the concentration of our copper sulfate. Our concentration is 0.62 moles per decimeter cubed. Yours might differ slightly from mine, depending on how well you've drawn your line of best fit. Well done. This brings us to the end of your virtual taster session for applied science. Hopefully it's given you a little insight into the subject and you've enjoyed it. I look forward to meeting you in September. Take care and thanks for taking part.